Once again, biology live class. This is number, where are we at? 5 out of 6. We have one more tomorrow, which is going to be a mini mock. If you are watching the recording before then I have published the mini paper, selected some questions. And the idea with this paper is to practice the exam technique that we have covered in the last couple of weeks. I haven't put many AO1 questions in there because the feedback I've had is people want to do more practice on the sort of more application-y type questions. Obviously, if you join my live classes ongoing between now and the end of the year, then you're going to get lots of exam technique practice along the way. Interesting, Jasmina. Okay, I will double check those links. It's going to be on this screen though, so I'm going to not be looking at the camera too much. Are you sure you're using? Well, let me. I'll pull the invite link from the room that I'm definitely in. So let's copy that, and I'll just double check that. Where do we want? I want this one and this one and this one. Let's just double check. I'm a few minutes. In fact, I'm not as early as I was yesterday. Okay, so the recordings, week 2. I'm just going to replace this link entirely. It should be the AQA link. I didn't look any different to me, but I'm replacing it anyway. Do you know what code's like? It's if there's a space in the wrong place, it doesn't work. How's that, Jasmina? Any difference? Okay, I've just swapped them out. Let me preview these pages and see what you guys are actually seeing. I'm just staring at code here, which is not my strong point. One of the things I've learned in the last few years, but I removed the button from the week 1. I think it looks kind of right. I have participants in the room, so definitely some of the links are working. Sarah is here. Let me just rearrange things. Hi, Sarah. Set those chat settings to panelists and attendees. Oh, it does work now. Okay, wonderful. Let's pull up the chat on that note. I'll change my chat settings. For those of you on YouTube, I'm going to be streaming to YouTube. I'm not going to be taking questions from the chat there. I'll keep half an eye on it. If you want to join and ask questions, then I'm doing so to all my students in the Zoom room, and the link to join that is inside of our courses. So there's a free trial from the Tailored Tutors website if you want to go across there. If you're watching on YouTube and this is useful stuff to you, then give us a thumbs up. It helps the algorithm share. I don't know what was wrong with that link. When I pasted it in, it didn't look like it was any different to the link that was there before. But yeah, thanks for letting me know. I changed it last night, so I was ready for today. The links are changing for all our different live classes, so I have to make sure that I double check them next time. All right, well, it's nearly past already, and I've got lots of questions lined up, and a few tricky ones coming up for sure. Jasmina's here. I think I see. Yeah, okay, let's go back to the YouTube chat just to make sure that I'm not missing out on anyone. No. Okay. Yeah. Refresh that page, click the link, come say hello. All right, so application questions submitted mostly by TT users. Pretty much everyone who sent me a question was an AQA student. So if you're sitting there thinking, oh, I do OCR, and you didn't send me a question, then that's probably why. I definitely was hunting around for some, but they are mostly going to be AQA questions. I suppose that is indicative of the fact that most students are AQA students in the country. That's just how it goes, that they're the biggest exam board for biology. Something is ringing, but I have no idea what it is. It's not my phone. I think it's a Zoom notification. It's very strange. Okay. What I'm going to do here is I'm just going to pull up, just in case I missed any last minute ones. All right. Well, let's jump in and get started. Hi, Kia. iPad. rearrange my screens when I screen share because Zoom's just like that. All right, okay. I'm just going to do a quick search over here. So what the? Okay, I think we're up and running. Let's resume that share. Let's jump straight in. Let's go. So here we have like a text. This question was submitted by, is it Itaza? I answer your questions all the time and I should know. You're here, so let me know in the chat. Is it Itaza? Well done for sending. This is a type of question. It's quite common for AQA. The other examples do give you blocks of text, and we're going to do another one later on that's like text plus diagram plus graph, and you have like all these different bits of info. So let's read it through together. Sizes of populations of normal intestinal bacteria. I'm going to just pick out things that might be relevant. So I'm going to say normal intestinal bacteria. So they're bacteria are commonly controlled by T cells, I'm thinking immune system, that are produced slowly, they're made slowly in small numbers by the immune system. Might be relevant. We know that T cells are made by the immune system, so I'm not going to highlight that. T cells do not normally survive for very long. Short-lived. So again, if you want to do that sort of exam technique, you can maybe short-lived. You can just like, these are the things that the mental notes that I'm sort of taking. As a result, they do not release large amounts of cytokines. Do not release large amounts of cytokines. Okay, let's not highlight that. Cytokines are chemicals that cause swelling of the intestines. So they do not produce large amounts. So we can infer that they do not do much swelling. Crohn's disease is a long-lasting disease that causes swelling in the lining of the intestines. Okay, so we're making lots of links here. It's believed Crohn's disease can be caused by a loss of tolerance, to normal intestinal bacteria, as shown by unusually large response of T cells. The response can be triggered by pathogenic bacteria in the intestines, so pathogenic bacteria, people with a genetic tendency towards Crohn's disease. Some people's Crohn's disease can be controlled by a drug called, I'm going to call it 5-ASA, that reduces swelling. Another drug called 6-MP may also be used. 6-MP inhibits an enzyme required to make adenine and guanine. This is effective because most cells can recycle nucleosides. T cells are not able to recycle nucleotides. Okay, use the information and your own knowledge. Okay, so this is actually any EdXL students here. Immune system is AS for AQA and OCR. It is an A-level topic for EdXL students. There will be more EdXL AS topics coming up. I should have maybe shuffled this one a little bit later on. Okay, the Crohn's disease symptom of swelling of the lining of the intestines could be triggered by pathogenic bacteria. Suggest how, 3 marks. So suggest how is like, what is it similar to? Again, I'm not going to do too much here. What's the actual topic of this question? So I'm going to throw that one open to you. In our carbs, our command word, we've done our actual topic of the question. What do we think here? Swelling triggered by pathogenic bacteria. It's basically immune system, right? It's like, what are the marking points here? I would say it's basically how can a normal response to a pathogenic bacteria cause swelling? T cells responding to pathogens. Yeah, we do need to use the text here to help us with this. So, okay, this is going to be a good exercise because I'm not going to put both on the screen at the same time. I'm going to give you 30 seconds to internalize this question. Make the question make sense to you. Reword it, rephrase it, reshuffle it. And then I'm going to move across to the text block, and then you can have that on your screen. And again, you can write your answers on paper. If you want, you can write them in the chat. So I'm going to be quiet another 10, 20 seconds. I'll get my timer open. And I'll give you 3 minutes to write your answer. Everyone ready? Ready? Have you got that question in your mind? This is where the question needs to be in your mind so that when you're answering it, it's coming from your brain and not you're not struggling to remember what the question is asking you. People ready? People got the question? Because you're going to just have 3 minutes of staring at the screen. Otherwise, I don't want you to start writing just yet because you're going to need to see the passage of text. Nobody has said that they're ready. Okay, Sarah. Thank you, Sarah. Excellent. Okay, so got to keep the command word the same. Suggest how the symptom of swelling could be caused by pathogenic bacteria in the intestines. Your 3 minutes for the R, the B, and the S of carbs. That's the bit we're doing now. Starts now. 3 minutes. It's my question analysis tool, Shania. I'll explain at the end of this question. Answer the question the best you can. Just give it a crack, Kia. Focus on the technique. One minute 20. Don't forget a self-check. Can you add in more key terms? I'll give you a 30-second warning for that. Just over 30 seconds. Short sentences, lots of key terms, simple facts. Concise sentences. Okay, time's up. Some people saying the Zoom link's not working. I just changed all the Zoom links. Refresh your page and you should be able to join the Zoom room. Yeah, I see you're in the class now anyway. So great. How many marks was this question worth? Excellent. I think everyone was in there with 3. If the question was I don't remember, then that's something we want to know every single time. What was our command word? Not an easy one. No, these are gonna be like that. Command word was suggest. Wonderful. Okay, so well, I'm going to come back to the question. So the symptom is swelling. what causes the swelling. So we can just do a bit of like, we've got these cytokines or these chemicals cause the swelling. So that's going to be down the end of our answer because we're going to end up with more of those. Why do we have more of those? Well, normal bacteria we don't respond to, but in the response to a pathogen, so we've basically got, I'm going to just summarize. So, oh, that's a highlighter. No good. Infection by pathogen. What's this pathogen going to be similar to? Going to have antigens similar to normal, in normal bacteria. If I'm trying to get my short sentences here, so you have a immune response. What I don't need to say immune response here. I can say T cells against the pathogens, and then the infection is over, but then the T cells respond to normal cells as well. Continue to make the chemicals which they tell us the name of, cytokines, and then more swelling, basically, which is linking it back to the symptom that is the question's all about. Okay, that would be my sort of bullet point exam technique demonstration. What do we get for some answers here? We got pathogens have a specific antigen. The phagocytes engulf the pathogen, present to the cell surface membrane. Complementary T helper cells bind to the antigen, stimulate the B cell, activate the T helper cells. Lots of T cells release cytokines, which cause the swelling. Okay, Sharu, that was like nice, but I think it's looking for the Crohn's is long lasting. So the pathogenic infection triggers the system to start responding to the normal bacteria after the pathogenic bacteria have been infected. Let's check the mark scheme. Okay, so the antigen needs to be in there on the pathogenic bacteria. It says assume that it's the pathogenic bacteria if you haven't stated it. Because the antigen is going to be similar on the pathogen and the normal bacteria. So the sort of, we're not responding to the normal bacteria. The pathogen comes along, we undergo the immune response, and it's suitably, the antigen is suitably similar for the normal bacteria to continue triggering the immune response. In other words, it's the protein has a similar tertiary structure. Causes more T cells to be produced. So an increase in T cells is basically the second marking point against the pathogen and the normal. Okay, so against the normal bacteria is what we need to say for the mark, as well as the pathogen. It's long lasting because the normal bacteria, I didn't say this is one I didn't get, do not die or live forever. I mean, live forever, live for longer, sorry. More cytokines or more chemicals causing swelling. So this was quite an accessible marking point because it was asking specifically about the swelling, and it said very clearly in the text that that was caused by these chemicals. You didn't have to name them. OCR expected to know this term, but no one else is. Okay, we've got quite a lot of things to get going through. So, okay, interesting. Suggest the meaning of genetic tendency to Crohn's disease, 2 marks. I'll go back and read line 10. This response can be triggered by pathogenic bacteria in the intestines of people with the genetic tendency towards Crohn's disease. And here we've got this word triggered. We've got Crohn's disease can be triggered. So that was, I guess, my clue here, that the infection is starting this whole process. That was, I guess, a key term. I didn't highlight it. I mean, it's hard to know what's going to be important before you read the question. This response can be triggered by pathogenic bacteria in the intestines with people with a genetic tendency to Crohn's disease. So what do we, I mean, this is a weird one. You're not likely to get it. I'm not gonna dwell on this one too much, but I'll give you 1 minute for this question. What is a genetic tendency? It's a weird question. I don't really know where to begin with this one, to be perfectly honestly. Megan, I must have said a million times that the amount of space is completely irrelevant, 100% irrelevant. This is the only thing that matters. 3 marks, 3 or 4 bullet points. Yeah, it's like they've chosen like a random number generator, but it's a random space generator. It's like how many answer lines to put in, make it up. It's okay. I'm just going to let people answer this question, Shania. Then I will answer a recap of carbs. I'm not going to dwell on this one. It's a very weird question. It's not something that's likely to keep sort of recycling. What are people's suggestions for genetic tendency? The likelihood to inherit disease. Yeah, we've got 2 marks though. So I think we have to mention the mutation or something in the genotype. Variation exists in the population. Some people have genes or alleles that mutations of alleles that cause them to be more likely to get Crohn's being inherited by their offspring, presence of alleles, or we can maybe talk about specific mutations. I think they're all in the right area. I don't know that I would have got 2 out of 2 for this one. In fact, you can watch what I did in the exam guide because I will have answered it. Yeah, we can talk about genes being switched on or switched off, potentially, all those things. It's a suggest question, right? So sensible suggestions are valid. Okay, I'll run through carbs just quickly after this, but some people have a mutation, allele, or a gene. Any of those that increase the chance, risk. It's more likely for them to have an unusually large T cell response that lowers the tolerance to normal intake. I don't think I would have. Again, it's linking back to the question, but I think I would have got this one. Tricky to get marking point 2. Crohn's disease. We've all talked about. Okay, the clue here, I suppose, is that this is talking about the name, and then it tells us what the cause is over here. So the genetic tendency is for what physically happens, and we give that thing that physically happens the name, the badge, the label of Crohn's disease. So I suppose if we could go a layer deeper, then we can always link it back to what's actually happening in the body rather than the name for what's happening. That makes sense? So a little bit of a lesson to be learned there. Carbs, very quickly, is my question analysis and answering tool. So this is the command word and how many marks the question is for. So that's the first thing you check every single time you see a question. This is the actual topic of the question. So sometimes it's not always obvious. This is R is review the key terms, because you want to do a little scan through your head of all the relevant key terms before you start writing, which is what happens here. We're going to use bullet points, bullet point your answers. The marks, as Megan just pointed out, is the only thing that tells you how many sentences to write, nothing to do with the amount of space available. S is, well, I did write my S twice. Self-check, which is basically, can you add more key terms? That's why we're practicing them today. Okay. Suggest 2 marks. Why? 5-ASA is only effective in controlling the swelling. Only effective in controlling the swelling. So they're calling effective by how much it controls swelling in some people. Okay, I'll go back to the text, but let's just, why is the 5-ASA drug only effective at controlling swelling in some people? Again, it's a suggest question, so we can go kind of off the map a little bit, but if we can tie it to something in the text. So we've got is a drug that reduces swelling. That's literally it. Some people. can be controlled by a drug that reduces swelling. Okay, so really the text I don't think is going to be particularly useful to us here. 1 minute, or suggestions just in the chat. Why might it not be effective in some people? Is in other words, probably the exam technique that I would give you here is like, what's the reverse? That might be sometimes easier to answer. Sarah, I think that is on the mark scheme from memory. We don't know how 5-ASA works. It could be a mutation in the shape of a protein that the 5-ASA attaches to. Allergy or adverse reaction. Maybe overreaction. So any suggestion as to why a drug does work on some people but does not work in others. Different amounts of swelling. Maybe some people have a crazy over response and the drug's just not able to cope with it. Yeah, maybe it's digested. I mean, you would probably have to assume, it's a good suggestion, Jasmina, that it digested before. If they said it was taken orally, then I think that's a valid suggestion. This is going to be done by doctors and scientists who wouldn't give a protein drug orally. They would give it intravenously. Yeah, here we're not so much talking about the pathogen. Here, the drug, what's the drug acting on? The drug is acting on the human, right? More than likely. So we probably can't talk about the pathogens, antigens so much. We can talk, this is probably a drug that's going to probably bind to a protein. So the binding site for this drug, its target obviously is a protein. It's got a tertiary structure. So maybe there's a mutation in this protein, which means that this binds less well or doesn't bind at all. These are all suggestions. Like, really, you should be given open range to just make a valid suggestion. But understanding this, if we're talking about the bacteria, we probably haven't understood the question. If we're talking about the human and how the human might respond, then totally fine. So various things. Some people might produce very large amounts of the cytokine, very large amounts of swelling. So the drug is just less effective because they've got more of the problem that the drug cannot control. Also accepting as a totally alternative answer, you can't mix and match these points over here. You have to go either these two or these two. Some people may be allergic, cannot tolerate the drug, so they cannot take it. It suggests questions frustrate the hell out of me because suggesting that there's a mutation in the binding target for the 5-ASA, quality response in terms of my opinion. Is it a mark? No, never will be, sadly. But just we've got to swallow the bitter pill that suggests questions. Okay, I want to pick up the pace a bit here because we've got a lot of questions to go through, and I think I want to just gain a little bit of a nugget of exam technique from each of these questions where we can. Suggest why the drug 6-MP can be used to control symptoms of Crohn's disease. So how can it control the symptoms? So we need to remind ourselves what the symptoms are. Swelling caused by cytokines. Do not include enzyme inhibition or protein synthesis in your answer. So do not. Okay, so let's go take a look. Get the question in your head. Suggest why the 6-MP drug can be used to control the symptoms. Another drug called 6-MP can also be used. This inhibits enzyme required to make adenine and guanine. This is effective because most cells can recycle nucleotides, but T cells are not able to do so. This is a good question, I think. This is a good application question. I'm going to give you 2 minutes. 1 minute to go. Last few seconds. Okay, if you've typed it, send it through. What's the actual topic of the question here? Would you say it's a bit of a mix match? Tazo, you've just gone straight in there with the first sentence, which basically tells us DNA replication or mitosis, cell division, mitosis. Yeah, specifically, it's more the DNA replication bit because that's when we're going to be using the nucleotides. Okay, so normal cells can recycle adenine and guanine. T cells cannot. So this is kind of a bit like a daisy chain. So suggest why, and again, we can rephrase this, a drug that inhibits, what does it do? That it inhibits the enzyme that produces, that makes, I've forgotten the word, that inhibits to make adenine and guanine. Yeah, so the T cells will not be able to reproduce their DNA, which means the T cells will not be able to divide, which means the T cells will not be able to produce cytokines, which means there will be less swelling. We don't really have to talk about other cells not being affected because they can recycle it, but T cells can't recycle their things. Therefore, if they can't produce adenine, guanine, they can't replicate their DNA, can't make new cells, fewer T cells, less cytokines, less swelling. And that's those stages are what the mark scheme is. I don't know exactly what the mark scheme is looking for. I don't cheat on these deliberately, so you get to see me like answering from my student perspective, not from my like, oh, I know what the mark scheme says. Okay, so we've got lack of adenine guanine will slow or stop DNA synthesis or replication in T cells. Let's go back to red actually. So slow or stop DNA replication in T cells. Affects T cells more because they cannot recycle these nucleotides, A and G, we probably should say here. Therefore, it slows the T cell or immune response, or you can say that there are fewer, again, marking point 3 is this one or this one. You can't get, it's just, yeah, either or. You don't get this one and this one. It's an or. So fewer cytokines, therefore less swelling. I think that was probably the cleanest, the most accessible question from that set. Any questions, any follow-up on that? Sharu, adenine, guanine not produced. T cells can't do DNA replication, so can't divide to produce new T cells, so less T cells productions, less cytokines, less swelling. Sharu, very nice short sentences, logical flow. Excellent. This is a good one here where sort of the actual topic of the question helps us, because it would be quite easy to talk about other things, but really the essence of this is the DNA replication, the mitosis kind of phase, as you well picked out. No questions? We're moving on. Next question. Okay, where are we at for time? Oh, geez. Okay, well, there's one. I'm going to come to this one, but it's a very similar question. I want, this is a real bitch of a question, and I definitely want to make sure we don't run out of time. So I'm going to do this one next. It is from an A-level paper, but it is about translocation and transport in plants. So it's an AS topic. I don't think much of this is actually A-level, but it is from an A-level paper. So I'll just have that in mind, and I'll bring it to the attention if I think there's something that's sort of out of reach for an AS student to be able to do. Let us begin. Scientists. So what do we know when we say scientists? Well, we can trust their method. It's not a student who may have cocked it up. It's a scientist. We trust them. Investigated the effect of heat treatment on mass transport in plants. They're barley. One species is what I'm thinking here. If we get asked a question, what does this apply to all the plants? Well, we've only done one species, so we don't know. They applied steam, so roughly 100 degrees or higher, to a short section of a leaf of the heat-treated plants. This area is shown with the arrows on figure 9. They did not steam leaves of the control plants. Okay, so there was no steam for the controls. They supplied carbon dioxide containing radioactively labeled carbon. So basically labeled carbon, we can tell where it is, shown by the rectangular boxes. So the carbon dioxide was applied here and here to each plant, which is above the steam point, right? If they were steamed. Okay, so I'm thinking this is triggering something in my brain, but I don't want to give you too big of a clue. What are plants using carbon dioxide for is the question. After 4 hours, so we don't know any long-term effects, but after 4 hours, they found the position of the radioactively labeled carbon. So where did the carbon go to? They recorded the water content of the plant leaf that was supplied with radioactively labeled carbon dioxide. Okay, so we've got water content and position of the sugar after 4 hours. Heat treated, control. If they were heat treated, that's where they were heat treated. We've got 0 hours, 4 hours. So this is what's happened here. Whereas in the normal plant, if you like, we can see the difference here. Plant from which the leaf was taken. So heat treated, group A. Control plant, group B. Water content of a leaf as a percentage of maximum. Obviously, okay, so if it's 50%, it's half of its maximum possible weight. Plus or minus 2 standard deviations are what these numbers are down here. I've put some planning on that. Ignore my blue numbers just for the time being. Okay. The scientists concluded that heat treatment damaged the phloem. Explain, okay, let's not circle. Explain how the results support the conclusion that heat treatment damages the phloem. So again, this conclusion, useless. Get it out of your head. What is the conclusion? Heat treatment damaged the phloem. So we're looking for reasons. How the results do support, well, it's not an evaluate, we damage the phloem. Okay, I think for here, I'm going to give you 1 minute. We need to be tight on time. What's the evidence that the time has begun? 1 minute. TW, which exam board are you in? Clicking the link in the course. Okay, so what's the clues here? I always talk about the clues in the question stem. Well, we've got CO2 up here, and in the heat treated plant, it stays up here. In the control plant, it ends up down in the roots. How is carbon dioxide getting from the leaf to the roots via translocation, via the phloem? So the carbon dioxide converted into sucrose in the light independent reaction, sucrose transported in the phloem by translocation from source to sink. In the experimental group, you can just say group A to save time if you want to, that no sugar goes below the heat treatment point. Therefore, the phloem has been damaged. We're explaining how the results support the conclusion. We could say that the phloem uses ATP and that living cells and that this heat treatment has killed them. But we're really explaining how the results support the conclusion, not giving the reasons why it hasn't worked. I think we should do these one at a time because I think, well, okay, so radioactively labeled carbon is converted into sugar or to sucrose during photosynthesis. I'd like to see light independent reaction there to be specific, because sometimes they are just ridiculously specific. And if you say photosynthesis and you mean light independent reaction, you don't get the mark. So let's be specific where we can. Mass flow or translocation happens in the phloem, or you can say movement of the sugar goes through the phloem, and it's only seen in the control or group B. Or, oh, what? Okay, another or. What's going on here? Why have they got, so this or is for marking point 2. That's one answer here. That's answer, I'm going to give them X, and this is answer Y. And then this is marking point 4, and you can have either that or that for marking point 4. Wow, okay, that's a confusing mark scheme right there. So that's option 1 is the top one there. Option 2 is movement in the flows requires living cells. Weirdly, they've given both versions of the answer here. Heat treatment damages the living cells, so transport in the phloem, only in the control. On the face of it, quite a nasty question. What are your reflections on that now that you've seen that mark scheme? Basically, there are different isotopes of carbon. Kia's asking what is the radioactive carbon dioxide? Normal carbon is carbon 12, but carbon 14 is a common isotope of carbon. So you can make CO2 from carbon 14. And so you can basically detect that this is different to normal carbon or normal carbon dioxide. It's like basically you've put a tag on it and you can see that version of the carbon dioxide. I'm just checking. Yeah, Jasmina, you're AQA, right? I'm pretty sure that we've got AQA students in the Zoom room. That the link is working. Just refresh the page in the course, and it should come straight through to the course. Yeah, Sharu, Jasmina. So the link, if you've had that page open for a while, the link was dead. Just refresh the page and then hit the link again. Should be good. Okay, let's not see the answer to the next question. Okay, so we've got scientists concluded that this heat treatment did not affect the xylem. Okay, so what are we doing here? We're explaining how the results support the conclusion that heat treatment did not affect the xylem. Okay, so we're giving reasons how the results support that. I've done Which data are we looking at here? What are we looking at to give us the answers for this? Table 4. What moves in the xylem? Water. So what are we looking at? We're looking at this. So what I've done here is I've taken the smaller number and I've added the 2 standard deviations to it. So that's what this value is here. And I've taken the bigger number and I've subtracted this value from it. And that's what this number is here, which will just save you a little bit of time on the calculations if you want to use those numbers, which I would highly recommend. If they give them to you, then okay, 1 minute. Okay, so is it useful to get my thoughts on this, or do you want to just check the mark scheme? It's obviously me quicker to go straight to the mark scheme, but if you want to see how I would sort of structure this in my bullet points, let me know if that's okay. So what I'm doing here, we've got the xylem. I'm thinking water. We're over to table 4. Well, we've done, do the standard deviations overlap? Yes, they do. That shows that there's no significant difference between the amount of water that's transported between the heat group A and group B. Therefore, we can conclude the xylem was not affected because water transport is happening as normal, or that there's no significant difference between the 2 groups. Okay, so Sharu, standard deviations for water content overlap. There is no significant difference in water content. So the xylem, which transports water by transpiration, transported water even in the heat-treated plant. So yeah, there's no difference between the groups. Therefore, the xylem was not affected. Kia, overlap of the standard deviations. There is no significance in the water content of levels of A and B. I'd say water moves in the xylem, just to state the obvious. I know it's something that sometimes it's quite difficult to do. Water content was not different because the standard deviations overlap. Water is therefore still being transported in the xylem to the leaf. Or you can say water movement is passive, so required, unaffected by heat treatment. Okay, excellent, everyone. I'm seeing improvements. And then the real stinger here, this one, I started, okay, so let's go through this together. The scientists then investigated the movement of iron ions, Fe3+, from soil to old and young leaves. So from soil, so the roots is what I'm thinking here, to the old and young leaves of heat-treated barley plants. We're still in barley and we're still heat treating. Compared that to leaves that were not heat treated. So same experiment basically, but different thing that we're looking at. Heat treatment was applied halfway up the leaves. Scientists determined the concentration of iron at the top and lower half of each leaf of each plant. Okay, so what I did here is I started sketching this out. So basically we've got old and young. We've got the iron is going in from the soil, right? So that's below ground. It's going up to the leaf. Heat treatment was made halfway up the leaf. So this is where I've put heat treatment. And obviously each one probably has its own control as well. And then they measure the concentration of iron at the top and lower halves. I mean, like, so then we test basically here, and for the sake of argument, here in the leaf, and we're looking for the iron. They're our test sites, and we're basically saying, is there Fe3 plus question mark, or how much is there at each of those sites? Okay, so that's where a sketch might help you. Like, it doesn't necessarily make perfect sense to me when I read it, that maybe just sketching it out is going to help us there. And we can try and decode what some of that's going on about. Okay, then we've got results here. And there's all sorts of shit happening here as well. We've got the ratio of iron concentration in the top half of the leaf to the lower half of the leaf. So what does that mean? So this is going to be like, where's pick a nice round number? Okay, let's call that 0.45 for the sake of argument right now. So that's at the top half of the leaf. It would be 0.45 to 1. If I'm writing my ratio super weird. So top to lower, it would be, yeah, 0.45 to 1. So there's more in the lower half than the top half. Are you following me so far? This is so far just like how I would go about decoding the question. Like, I'm just making sense of what the hell they've given me here. This is an unusual one. Has anyone sort of top half to lower half? So the 2 is this. So the top half is the 0 point whatever from the graph, and then it's a ratio, so it's to 1. All right. Now we understand what's going on. What's the question? What can you conclude about the movement of iron in barley plants? Use all the information provided. So here, my question is that all of the information, as in all of this crap as well? Probably. It does say all. I don't know how much of that is relevant, just FYI. So what can you conclude? What are the conclusions? So this is AO3 drawing conclusions about the movement of iron in barley plants. Okay, remind me before we answer this, what's our technique for these sort of like conclusion style questions? We did this, I think, in the answering technique lesson a week ago today. What's our technique here? Yeah, we can. Typically advantages, disadvantages. Yeah, we totally can do that. We can, when under my general points, generally advantages, disadvantages are going to be evaluate questions. This is a conclusion question, but it's similar. Points for and against, things from the graph, pieces of info and an explanation. Yeah. So the technique here, I want individual nuggets was the word that I came to last week. Yeah. So like when we get these like evaluation of investigation questions, we're looking for as many short sentences. You complete the sentence in your brain before you write it down. This should not be a flowing passage of text that comes all the way down the page here. That is not the technique. Technique is fact 1, full stop. Fact 2, full stop. Fact 3, full stop. As many as you can, and then basically evaluate the practical. So criticize the prac. What are some example criticisms that we can give of any experiment? Obviously, it depends what they've done, but like general criticisms. Sample size, duration, stats test, repeats, only for one species. Here, they've said, what can we conclude about barley plants? So we can't say they haven't tested other species because we're only being asked about correlation versus causation. Exactly. Okay, so excellent. You guys, my Padawans, are learning very quickly. Okay, so this is what I would finish with once I've done all the facts that I can get. other factors that could have been controlled. Yeah, here it was done by scientists, so we don't want to spend too much time on that. But if you've got genuine criticisms or evaluation of the practical points, do include them. And I generally just tack them on at the end. I think of all the answers I can give, short sentences. Okay, shall we do this one together? This is a tricky one. Okay, I don't know why that eraser is being a pain in the butt. All right, so I'm now looking for facts that we can conclude about the movement of iron, right? So the data over here, we know from this experiment that the phloem is affected and the xylem is not. That's basically the conclusion we can make that's relevant here. All right, let's give this a nudge. What can we conclude? Single one sentence difference that we're looking for here. We can do old versus young. We can do heat treated versus not heat treated. Okay, heat treatment affects young leaves more than old leaves. Full stop. We say more movement in younger leaves. What? Anyone else having connectivity issues, or is it Jasmine is? Yeah, my internet stable's pretty connection. My internet connection is pretty stable. Okay, you could always, yeah, maybe crash and rejoin. Okay, so young leaves affected more than old leaves. Heat treatment affects young leaves more than old leaves. What can we deduce? No, it's a tough one. What could be different then? Young leaves are growing, they're forming new tissues. There's an interesting point. That's quite a deep application point, the one that I've just thought of. If it's young, it's growing, it's got I don't want to give it to you. Any more facts that we can give here? We've got one so far. Yeah, the xylem has already grown in old leaves. That's potentially a difference. Yeah, potentially we could flip that and say the xylem is still potentially living in young leaves. It's not died yet. It's not been lignified. Young leaves not fully developed. It would be nice to connect that to the xylem possibly still having living tissue. Higher metabolic rate in young leaves, so more active transport. Yeah, but can we, what does the data tell us? Let's what can we say here? We can compare this with this with this. We can do young versus old. We can do the two grays. We can do the two whites. Heat treated have a higher ratio of iron in the tip than the lower leaf than untreated leaves. The lower the amount of iron in the top, the more likely it is the plant uses the phloem for transporting it there. Yeah, because the heat treatment has caused the reduction. How is iron likely to be moving in the old leaf? Iron, it's an ion. I actually don't know. I'd assume it's actually traveling in the xylem. If you'd have asked me before this question, I'd have said, yeah, probably in the xylem. It's a dissolved ion, but it's obviously it has some influence in the phloem. So we could maybe say in old plants, iron is moving in the xylem. In young plants, oh, it's so tricky. I think what's actually happening here is that the xylem is not dead yet because the young plant is growing, and therefore the xylem is being heat treated, and it's the xylem that's being affected, and that's what's causing it. That is like quite a deduction, if that's the acceptable answer. But that will be one of many acceptable answers, I would assume. Or you could, yeah, the old one can also travel in the phloem. Probably not xylem traveling, the iron traveling in the phloem, because both are heat treated, right? So compared to the normal, in the old plant, iron is still reaching the tip normally. Yeah, that's a statement. Whereas in the young leaf, heat treatment causes a big reduction in the amount of iron reaching the tip of the leaf. Let's go straight to the mark scheme so we can see what some of these things are. Oh, and a criticism, I would say, if I don't glance down there and I don't, what would I say? We can't talk about only barley plants. There was no stats test done. We didn't get any stats test on that. That would probably be a simple one. Oh, yeah, it's in there. Freebie, a little freebie mark at the end there. Huh? You can thank me that. Even if you just write no stats test, you get a mark here. Okay, heat treatment has a greater effect on young than old. Boom, that was the first one in there. Nice, simple sentence. Relatively easy one to deduct from the data. Heat treatment damages the phloem. Okay, that's a conclusion, I suppose, from all of the data. Iron moves up the leaf, up the plant of the leaf, from the roots to the end of the leaf tip. Wow, I would never have thought to write that, to be honest, but it's a mark on there. It suggests that iron is transported in the xylem in the older leaf because basically the two bars were the same in the heat treated versus the non-heat treated. In the young leaf, some xylem, some travel in some iron in the xylem, but still some reaches the top of the plant. In fact, that's a weird garbled marking point if ever I've seen one. This suggests that iron is mostly transported in the phloem in the younger leaf because of the big difference. Or you can say the xylem is damaged in the younger leaf, the xylem is still alive in the younger leaf. That was the point that I was getting close towards. The higher ratio of iron in all untreated old leaves than all untreated young leaves. High ratio of iron in the old leaves, get rid of this, in the old leaves than the young leaves. So basically, these bars are taller than both of these bars. Again, so these are, you can put in as many of these short sentences as you want, all the ratios, so there is less iron at the top of the leaf than the lower leaf, and that's we get from the ratio. So there's roughly, well, if we were up to 0.5, there'd be half as much in the top than there was at the bottom, because we know ratios are 0.5 to 1, means that you've got half here for a full dose here. You don't get the reference to the xylem. Which one here? Marking point 6, which marking point, Shad? Okay, so it suggests that iron is mostly transported in the phloem in the young leaf because it's massively affected. There's a big difference between the untreated and the treated. Or you can say that the xylem, it's a young plant. So at some point, the xylem must be alive in order to grow. That vascular, in a mature plant, the xylem is lignified. It's like a plastic wrap. It causes the cells to die because they have no exchange. They need to be waterproof. So in old plants, the xylem is dead. But at some stage, the xylem must be alive. So in these young plants, maybe the xylem is still living, and therefore the heat treatment damages both the xylem and the phloem in young plants, where it doesn't affect old plants because the xylem is already dead in the old plants. Does that now make sense? Definitely a tricky one. Again, what I really wanted to focus on here was the method. Okay, so let's just go back to here. We get what can you conclude? Anything that you can see as being a fact, any single fact. We don't need to like write. Okay, I'm just struggling so hard. Do you see how writing a long flowing paragraph about all the different things that you have seen and like what your general conclusions are, do you see how that is not useful? Can I get a yes or a no to that? Do you see how like as many individual single factual conclusions that you can draw, like the simple one was like old plants are less have more iron in their in their shoot tip than young plants, like you can put in as many as you want, and the more points you get in, the more likely you are to get answers. With these kinds of questions, you're not ever going to get deducted for writing facts, but if you write more points, fewer words in each point is the way. But they're not easy. I'm not suggesting that now you should walk away and get 100%. What I'm suggesting is you need to practice this method in your own time, do a bunch of past papers, and then see you're going to incrementally pick a few more. And then when you check the mark schemes, it's like, oh yeah, definitely could have done that. Definitely could have got that. Or these general points as well. Oh, no stats test done. Bing, free mark. Some of these marking points are quite accessible, but you could approach this and then to an untrained person, it's like, holy shit, I've got to write a page of notes here. I've got to write all of this, and they're going to start writing here and they're not going to engage their brain again until they get to like down here and think, well, I've written loads. Probably that's all right. Not the way. Everything is simpler as short sentences. Yeah, that's carbs for you. Get the key terms in your head first, or in this case, I suppose the review the key terms is like review the data rather than the key terms. You still want to get key terms in there, but it's like review the info. But you do have to practice it. I'm not going to pretend that I have done millions of versions and I would probably have still got, I probably would have got 3 out of 4 here. I bet there was at least one marking point that I wouldn't have got. Definitely. In fact, you can literally go back and watch me answer it with for the first time I ever saw this question and see what I got. All right. I have put the mini mock up for, there's another vaccination question. I don't think I was going to do this as another sketch example. That's in the mock over there, so I'm not going to go into that one. Okay, I'm going to just, I'm going to show you. Oh yeah, I've got to switch window first. Okay, so in the live classes, I'm in the edXL one here, but I've got all of them open here because I just had to change the links. This is the question paper for tomorrow. I didn't put too many questions in there. I forget how many marks it is. But I think there's 3 or 4 full questions like we've done today. We've followed a question through all the way through. So have a go at that. Ideally have a crack before tomorrow, even if you just do it double speed, brainstorm, actual topic of the question, command word and marks, bullet point your answers, just first thing that comes to mind, and then we'll be able to have something to compare against when we do the live class tomorrow. So any questions that were submitted that I basically didn't get to are in the mock. I chose some for the mock and I chose some for today's lesson. So it shouldn't take too long. Do what you can. I know you guys are busy right now. Some of you are back at school or college. Some of you are just revising with TT revision method. That probably takes priority over this, to be honest. But this will be a nice little exercise for us to see if we can pull it all together. What I really like you to do when you're doing this question paper is try and apply as much technique as you can. And And then when we mark it tomorrow and we go through the questions, we can see, can we do differently? So summarizing some of the exam technique that we've done. Carbs. If you're not sure what that means, it's in the exam guide in the exam technique section if you need that, or you can watch the live class on question analysis from last week. We can write in short sentences. We can try and decode the words in the question. So today we had the swelling. So we knew the swelling was caused by the cytokine. So like connecting the clues in the question to the science underneath it. We've got getting the question clear in your mind before you start writing, knowing each sentence, compose each sentence in your head before you start it, and you don't just continue writing the next sentence. That's why I use bullets. Bullets is like nugget, fact, bullet, full stop, next nugget, fact or bullet. It's not flowing prose, because otherwise we just write waffle. You get pure 100% unadulterated waffle when you do that, and that is not a great way of picking up marks in biology. Rephrasing the question, if you can reword some of those things so that it makes more sense to you, again, all part of getting that question crystal clear in your mind, so that when you're writing your points, it's coming from your brain and you're not cross-referencing, cross-checking. You should always know the command word. You should always know how many marks it is. If we're evaluating and if practical, we can throw in some of the criticisms. Suggest questions are usually more simple than you think. It's what is this similar to that I do know about? Don't focus on like, oh, it's asking me about this weird and wonderful thing that I've never come across before. What is it similar to? Brainstorm those key terms, run through it. Remember that you are not going to get 100%, and that is okay. That suggest question earlier about I think it was the T-cell drug, the Crohn's disease drug. Like, my suggestion, as far as I'm concerned, was a good suggestion. No marks. So we just have to literally bite the bullet on that one. A lot of it, Sharu, yeah. In fact, some of the work I'm going to do over the summer is speaking to Rich and see how much of it can be applied, and how much of it maybe isn't the best technique to apply to chemistry. I would think for evaluating experiments and stuff like that, it would be, broadly speaking, very similar. So Daph saying, getting better at suggest questions really helps to think what it's similar to. And yeah, okay. So that mindset, yeah, they're not expecting you to know stuff outside the spec. They can only expect you to know things inside the spec. So we're looking for that similarity and then going from there. 100%, the recording will be available. I'll put today's recording up when it's processed in a couple of hours, so it'll be there for tomorrow morning. And obviously tomorrow's recording will be up same time the next day. Good work, everyone. Marima, are you with me today? Where you did suggest a question that is not in the mark. You are here. If I don't get to it by the end of tomorrow, then I will record a video on that Alzheimer's question and I'll talk you through it. It's only 5, 6, 7, 8, 9, 10. It's 10 marks total. So if I don't get to it tomorrow, just give me a reminder about the Alzheimer's question that you suggested, and I'll make sure that I've gone through it with you. Have a go at the mock. If you don't have much time, just pick a couple of questions, apply the technique, practice, practice, practice. See you tomorrow. Bye.